We're going to look at primary and secondary active transport in this lecture. Remember, in active transport, molecules are going to be moving up the gradient, so going from low to high concentration. And so in order to go up the gradient, we do need to put energy in to do that. We're going to be moving hydrophilic molecules using a pump, and this transport protein is going to be very specific to which molecules it's going to move across the membrane. It's also only going to be going one direction, so it's not like the carrier protein in facilitated diffusion where they could go either direction. So it'll just go one direction, and the pumps can also become saturated just like the carrier proteins in facilitated diffusion. There's two different types of active transport. We have primary active transport, which is going to be directly using energy. And then we have secondary active transport, which is going to be using energy indirectly. Let's take a look at primary active transport with our sodium-potassium pump first. We're going to be talking about this a lot this semester. And let's draw our cell with our sodium and potassium concentration gradients. So sodium is much higher outside the cell than inside. And potassium is just the opposite. It's higher inside the cell than outside. And here's our pump, our transport protein. And we're going to be moving 3 sodium ions out of the cell. And then we're going to be moving 2 potassium ions into the cell. And notice that when we're moving both the sodium and the potassium, each one is going up the concentration gradient. So sodium is going from low to high, and then potassium is also going from low to high concentration. And in order to move up the gradient, we're going to be directly using energy, some ATP, to power this. And this picture from your book just shows the binding sites for the sodium and the potassium. So you can see that we've got 3 places for 3 sodium ions, and then the ATP will chop off a phosphate group. That'll release the energy as we phosphorylate that sodium-potassium pump. That'll cause it to change shape and then move the 3 sodium out of the cell. And then the phosphate group will then come off as the 2 potassium bind to their binding sites in the pump, and then that will cause the pump to change shape, and it will flip to the other side and move the 2 potassium into the cell. Here's an animation showing the sodium-potassium pump, and you can see the concentration gradient for sodium. This red thing is ATP, so when we phosphorylate the pump, we're going to move 3 sodium out of the cell. And now we've got 2 potassium binding to the pump. Phosphate group falls off, and that'll change the shape of the pump again and move 2 potassium in. And you can see that we're moving the 3 sodium out of the cell, up the gradient, going from low to high. And then we're moving 2 potassium ions into the cell, also up the gradient, going from low to high potassium. So the sodium-potassium pump is really important for a couple of reasons. It's what creates the concentration gradients. So this is why we have sodium much higher outside the cell than we do inside, because our pump keeps moving sodium out of the cell. So we move 3 sodium out, and if we keep moving sodium out of the cell, the sodium is going to be lower inside than it is outside. And then we're also going to be establishing our potassium concentration gradient because we keep pumping potassium into the cell, and that's why we have a lot more potassium inside the cell than we do outside. And these concentration gradients allow secondary active transport to work, which we're going to talk about next. And this also helps establish our negative membrane potential that we need for the nerves and muscles to work. And that's because you're moving an uneven number of ions with the sodium-potassium pumps, you're moving 3 sodiums out, so 3 positive charges out of the cell, and you're only moving 2 potassiums or 2 positive charges into the cell. So if you have 3 pluses leaving and 2 pluses coming in to replace them, that's uneven. So we have more positive charges leaving than coming in, and so eventually we're going to end up with a negative membrane potential, and we'll talk about this later, but we get down to minus 70 millivolts with that. And the other important thing is, because our sodium-potassium pump is a form of primary active transport and directly using ATP, as we break the ATP down, that's going to generate some heat. And that's where a lot of our body heat comes from, is from our sodium-potassium pumps. Here's just another example of primary active transport. This one is a proton potassium pump that we find in the stomach. And you can see that we've got in our box here, if you take a look at the hydrogen ions, they're going to be moving from low to high concentration. And then you can see potassium is also moving from low to high concentration. So both are moving uphill of their gradients, and this is how we get the hydrogen ions out into the stomach lumen, which will then be combining with the chloride ions to produce the hydrochloric acid, the stomach acid. And obviously we do need energy to do this, because we're moving up the gradient with both the hydrogen ions and the potassium ions, so this does require energy. And here's just another example of primary active transport. This is a calcium pump, which we're going to be talking about when we get into the muscles, because calcium is going to be used to help contract the muscles. And once we're done contracting, we need to get rid of the calcium. So you can see that we're going to be moving our calcium ions from low concentration to high concentration. So we're going from low to high, we're going to be going up the gradient. And you can also see in our picture that we're using ATP directly to power this, so this is primary active transport. In secondary active transport, we're going to be using energy indirectly. And the way that you recognize secondary active transport is that one substance is going to be moving down its gradient and the other one's going to be going uphill. So if you see a pump, one going down the gradient and then the other one going up the gradient, then you know it's got to be secondary active transport. With secondary active transport, we have 2 terms that we use, and these are only used with secondary active transport. You're not going to see these used with primary active transport at all. So the first one is cotransport, also called symport. So co and also sym both mean together. So we're going to be transporting 2 molecules in the same direction. So for example, we're going to be transporting both of them into the cell. And then countertransport or antiport, we're going to be transporting the 2 substances in opposite directions. So one's going to go into the cell and one goes out of the cells. All right, so let's go ahead and we'll draw some pictures. And let's take a look at, maybe we're in the intestinal lumen, and we've got an intestinal cell here. And we want to move glucose into our intestinal cell, because then we can move it to the other side and get it into the blood. And let's go ahead and put in our concentration gradients here. So we've got sodium higher outside the cell than inside, and then the glucose, because we keep moving it into the intestinal cell, is going to build up in the intestinal cell until we move it out on the other side. So, and we'll get to that later. And how we get our sodium concentration gradient is we're going to use our sodium-potassium pump. So remember that we're going to be pumping 3 sodium ions out of the cell and 2 potassium ions into the cell. and we need ATP to do that. So this sodium-potassium pump, that's primary active transport. And having this sodium-potassium pump is going to keep the sodium ions low inside the cell because we're going to keep pumping them out. And that's going to make the sodium higher outside the cell than inside. And that's going to allow our secondary active transport to work. So with this pump, we're going to be moving sodium into the cell. And at the same time that this transport protein grabs the sodium, it's also going to grab a glucose ion as well. And so it ends up moving both of them into the cell. And if you look at the concentration gradients, you can see that sodium is going from high to low, so it's going down its gradient. And then glucose is going up its gradient. It's going from low to high concentration. So we know this is secondary active transport because we've got one molecule sodium going downhill and the other molecule glucose going up the gradient. So this will be secondary active transport. And the other thing that we don't see in our picture with secondary active transport is that we don't have any ATP that we're using here. So we're going to be indirectly using energy because we had to use energy over here with primary active transport to establish our sodium concentration gradient, but we're not directly using ATP with our pump here on secondary active transport. If we blocked the sodium-potassium pump, if that was no longer working and we could no longer pump the sodium ions out of the cell, sodium ions would come in and then eventually they're going to be the same as what's on the outside of the cell. And then that would stop secondary active transport from working and that would shut down. All right, so let's take a look at another example. So let's take a look in our kidneys, and we're going to use sodium again. So that's higher outside the cell than inside, and how we got the sodium concentration gradient was from our sodium-potassium pump. So we keep moving 3 sodium out, 2 potassium in. We're directly using ATP there, so we've got our primary active transport. And that's going to set up our sodium concentration gradient. And then we're also going to be moving some hydrogen ions. So this pump is going to take sodium and move sodium down the gradient going from high to low concentration. And at the same time that it grabs sodium, it's also going to grab a hydrogen ion. And we're going to move the hydrogen ion out of the cell. And so we're going to move it from low to high concentration. So again, this is secondary active transport because we've got one molecule, our sodium ion, going down the gradient, and then the other molecule, our hydrogen ion, is going up its gradient. And again, notice that there's no ATP in this picture. We're going to be indirectly using energy. We had to use energy with primary active transport to set up the sodium concentration gradient, which allows secondary active transport to work. Okay, and then if we take a look at these and apply our other names that we use with secondary active transport, so this secondary active transport that's up on top, we're moving both the glucose and the sodium ion. They're both going into the cell. So we would also call this symport or cotransport. And then the one on the bottom, because sodium is going into the cell and hydrogen ions are moving out of the cell, we could also call this one antiport or countertransport. All right, so we'll just take a look here. This is what we had drawn on the previous picture, and I've just got a little animation here. So if we have sodium going down its gradient, and we can see that in our picture, sodium's moving from high to low concentration. And then we've got glucose moving from low to high, so it's going up the gradient. So if you got one going down the gradient, one going up the gradient, then we know this is secondary active transport. And then we can assign the name, is it symport or antiport, by looking if both things are moving into the cell or if we have one moving in or out. And then if we look at our little animation here, you can see the sodium and the glucose are both moving into the cell. So this will be an example of cotransport or symport. And then this next one was the other one we drew with the sodium and the hydrogen ions. And we know it's secondary active transport because sodium is going down its gradient, going from high to low concentration. And then hydrogen ions are being moved up their gradient, going from low to high. And if we look at our animation here, you can see that sodium's going in, but hydrogen ions are moving out. So we could also call this one countertransport or antiport. And then this is a really good table to study for the quiz and the exam. So it has all of the different types of membrane transport that we've talked about so far. And I want you to know if you're going up or down the gradient, so make sure you pay attention to that. So remember, all the passive transport, anything with diffusion in the name, is going to go down the gradient. And then primary and secondary active transport will go up the gradient. And then the only one that does not require a transport protein would be your simple diffusion, and everything else is going to have some sort of a transport protein. And then make sure you know which ones use energy and which ones don't. Obviously, only the active transport will require energy and everything else will be passive. And then I'm not going to ask you, well, the energy source, just know that primary active transport, we're directly using energy, and then secondary active transport, we're indirectly using energy. And then also, do things become saturated or not? Simple diffusion, because you're not using a transport protein, you're not going to become saturated. So the bigger the concentration gradient, the more molecules you can move across. Whereas all the others, using a transport protein, they can become saturated, and they get to a point where you can't move any other molecules any faster across the membrane unless you put out more transport proteins. And then if you're using a transport protein, it's going to be specific for what it transports across. And then notice that simple diffusion will transport those hydrophobic non-polar molecules. And then also we can add here our small polar molecules like water as well. So they can go across by simple diffusion. And then everything else is going to be transporting hydrophilic molecules. You don't need to memorize this bottom row here, but just know that you're transporting hydrophilic things. And then we'll be talking about this later, but basically the cell is just a system of pumps and leaks. Okay, so for example, we'll talk about some sodium leak channels later. So sodium will move down through its sodium ion channel, okay, which is always open because it's a leak channel, no gates on it. And so sodium will move into the cell, and as soon as sodium comes in, it gets pumped back out again. Okay, and then the same thing with potassium. It gets pumped into the cell by the sodium-potassium pump, and then it will leak back out through the potassium leak channel.